Welcome to our lecture on puberty. This presentation was adapted from the lecture of Dr. John J. Parrish. This presentation aims to be able to describe the endocrinology behind the male and the female puberty of domestic animals. So what is puberty? In reproductive physiology, puberty is defined as the ability to accomplish reproduction successfully. It is important to note that puberty is a process and not a single event. So an animal that will undergo puberty will have to, for example, when the animal will be mated, it can undergo conception, it can undergo gestation, and right after that, after a certain gestation period, it will undergo parturition, nursing of the young, lactation, and after that there will be weaning. And that is the time that the animal will then undergo another cycle of estrus. So this is the reason why puberty is not just a single event, but it is really a process. It comes from the Latin word pubescere, meaning to be covered with hair. Of course, this definition applies to the development of hairs in the pubic area, armpits, and legs, particularly in humans. But of course, this does not apply in other animals. The fundamental requirement for the pubertal onset is the secretion of the hormone gonadotropin-releasing hormone, or the GnRH, which comes from the hypothalamus, to stimulate the gonadotropin release from the anterior pituitary. Of course, these gonadotropins include FSH and LH. Before discussing puberty, it is important to understand that there are fundamental differences in the hypothalamus of the male and the female animal. The GnRH comes from two regions in the hypothalamus. We have the surge center and the tonic center. So in this diagram, the tonic center is colored green, while the surge center is colored pink. The surge and the tonic centers are important because they are composed of groups of hypothalamic nuclei that influence reproduction. The female has both the surge center and the tonic center, whereas for the male, it only has the tonic center. This slide will demonstrate why the male brain does not develop a surge center. So of course, we have here the testes, the prenatal testes, the brain. We have here also the hypothalamus, and the red one here is the blood-brain barrier. So in the prenatal development in the male, the fetal testes will produce the hormone testosterone, and it defeminizes the brain. In order for testosterone to defeminize the hypothalamus, it must first cross the blood-brain barrier. So of course, the testosterone is a steroid hormone. It is lipid soluble. It will readily cross the blood-brain barrier and enter the brain. And within the brain, the testosterone will be aromatized into or converted into estradiol or E2. The E2 here is the one that is responsible for the surge center to not develop. That is why in the male, the only present is the surge center, and it is the one that is responsible for the basal production of the GnRH. For the female brain, the estrogen or the estradiol is produced from the fetal ovary and the placenta. In the female, the E2 is unable to cross the blood-brain barrier and gain access to the hypothalamus. This is because the E2 binds with alpha-fetoprotein and it prevents it from crossing the blood-brain barrier. Hence, the E2 cannot affect the hypothalamus. So thus the hypothalamus is feminized and the surge center develops in the female. So hence we can say that the hypothalamus is inherently female. This diagram shows the fundamental differences in the endocrine profiles of the post-pubertal male and female in domestic animals. So of course the y-axis represents the hormone level, while the x-axis represents the time. This is the top portion. We have the female patterns, pulse pattern. Of course, in the female, we have the surge and the tonic center. And for the male, we only have the tonic center. So as you can see in the diagram, so of course, as you can see in the male, so the LH does not surge in the male animals. So only it maintains a tonic secretion of LH day in, day out episodic pattern. So when we are going to enlarge this point here, we can say we can see that the hormone that is present now in the male in this diagram is the LH, or the luteinizing hormone. Of course, the LH is important for the secretion of the androgen. So that is why we have here the T in every low amplitude surge of the LH luteinizing hormone from the anterior pituitary. We can also see here a surge in the, of course, a tonic, a tonic increase in the testosterone coming from the testes. That is because LH is important for the production of testosterone in the male animal. Of course, it is very different from that of the female, no, because uh, right after several days, there is a surge. There are surges in the luteinizing hormone as seen in the diagram. Now, so of course, before the LH surge, we also have the surge of the estrogen or the E2. This is important in order to um, this is important as a positive feedback for for the production of the GnRH. And when there is an increase in the GnRH, it can also simulate the production of the LH from the gonadotropes. So of course, this is true for the female animals, and the interval between one LH surge to the other is the estrous cycle period. For example, in the cow, this is about 21 days. So again, for the female, the LH is important for ovulation to occur and for the production of the hormones in the female animals. When we are going to enlarge that, of course, we can see that we also have here the same as in the male animal. We also have here the presence of the luteinizing hormone in the female animal. Of course, these differences are due to the tonic center and the surge center in the female. So we have here the surge. The surge center is responsible for the preovulatory LH surge. Now that is important for ovulation. For the male animal, so it is maintained in a basal level. So this is due to the tonic center. When we are going to again to enlarge this portion here, the females have a high amplitude. Again, this is a high amplitude preovulatory episodes of LH once every several weeks, and basal pulsatile episodes between the large preovulatory surges. So ito yung LH surges. Of course, that is different from that of the male animal, because in the male animal we have small LH episodes that occur every 2 to 6 hours as shown in the diagram. Testosterone is secreted soon after each LH episode as seen in the diagram. Criteria to define puberty in female animals. So the first criteria used is the age at first estrus. So this is the age that the female becomes sexually receptive and displays estrus. Of course, the age at first estrus may not reflect the true acquisition of puberty in female animals. That is very important to remember. In the female animal, of course, in the first onset of puberty, we also have the phenomenon known as the silent ovulation. When we say silent ovulation, this is the first ovulation that is not accompanied by behavioral signs, such as this is usually seen in heifers and ewes. So um, at this particular ovulation, so it is not recommended that the animal will be bred at this point. The next criteria is the age at first ovulation. So this can be confirmed by manually by rectal palpation or visually by the use of ultrasonographic imaging validation. The next criteria is the age at which the female can support pregnancy without deleterious effects. This is considered to be the most applicable criteria in all domestic animals as well as in humans. The acquisition of a threshold body size is important in controlling the onset of puberty in female animals. So this is because the goal for food-producing species is to generate the highest possible number of offspring in the shortest time interval without compromising the well-being of the dam or the neonate. In order to do this, of course, the dam or the female animal must be able to meet a certain threshold body size. These are the criteria to define puberty in the male animals. Number one is the age when behavioral traits are expressed. So this includes mounting and erection. We also have the age at first ejaculation, age when the spermatozoa first appear in the ejaculate. So the male acquires the ability to produce the seminal fluid and to ejaculate before sperm are available to be ejaculated. And the last is the age when the ejaculate contains a threshold number of spermatozoa. So this is considered to be the the most valid the most valid criterion for puberty in the male, since it defines the ability to provide enough spermatozoa for fertilization or for successful fertilization to occur. For the quiz, why does the hypothalamus in the male not develop a surge center? The testosterone is secreted by the fetal testes. It is converted to estrogen in the brain, and then the estrogen prevents the surge center from the development in the hypothalamus. We also have now in the female, estrogen is secreted by the placenta in the ovary. That estrogen does not reach the brain due to the binding to the alpha fetoprotein in the brain. The surge center does not see estrogen and therefore develops in the female animal. These are the factors that are related to the onset of puberty in male and female animals. So of course, first is the hormonal. These are, for example, the GnRH hormones. We also have the genetics. Of course, we have the nutritional status of the animal and the environment. For the hormonal, we have the GnRH. Now from the hypothalamus, this is the hormone that is important for the onset of puberty in both males and female animals. The GnRH is very important because the pulses determine the onset of puberty. So prepubertal or before puberty, in terms of the GnRH release, there is a minimal GnRH release, and when the GnRH is minimal, the FSH and LH would also be low. Because of this, there is also minimal to no folliculogenesis or spermatogenesis. For the pubertal stage, there is an increase in the pulse frequency and the amplitude of the GnRH release, as of course seen in the female. There is an increase in the FSH and the LH pulses, and these hormones are important for folliculogenesis and spermatogenesis to occur. For the hormonal changes that is seen in the female animal, this is the hypothalamic secretion of the gonadotropin-releasing hormone before puberty. So before puberty in both the female and the male animal, the GnRH neurons in both the tonic center and the surge centers release low amplitude and low frequency pulses of GnRH as shown in the diagram. So after puberty in the female animal, the tonic center as shown in this diagram. So the tonic center will control the basal levels, now as shown in this diagram, the basal levels of GnRH, but they are considered to be higher than in the prepubertal female because the pulse frequency increases. We also have here the surge center, and the surge center controls the preovulatory surge of GnRH. Again, the preovulatory surge is important for ovulation. The male does not develop a surge center. This is a comparison of the hypothalamic secretion of the GnRH before puberty in the female and the male animal, and after puberty in the female animal. So of course, after puberty, we have the surge center that induces the preovulatory surge for the female animal. In addition, we also have the tonic center, which is higher compared to that of the prepubertal female animal. The surge center is sensitive to positive feedback and will then release a high amplitude, high frequency pulse of GnRH for a short period of time when estrogen has reached a certain level. In this diagram, the GnRH surge in the female animal is compared with that of a tap of water. So when there is a surge burst, consider the tap of water to be fully flowing for a short period of time, so as shown in the diagram. So there is a surge in the GnRH as well as, of course, in the LH that is known as the preovulatory surge. When we are going to compare that with the tonic center, of course, the flow of the water in the tonic center is considered to be a few drops only, which represents the tonic basal layer secretion of the GnRH at this time. This is a diagram of the LH frequency before and after puberty. So this number here represents the months before puberty and the months after puberty. So months before puberty, we can see here that tonic LH pulse begins to increase minimally as shown in the diagram. At puberty, there is a surge in LH, a surge in the LH surge or GnRH. After puberty, it recurs at repetitive days, now as shown in the diagram. Of course, these repetitions in the um, preovulatory surge or the LH surge represents the estrous cycle. For example, for cows, mare and sow, this surge here occurs every 21 days. For sheep, this is about 17 days, now during estrous surge. The surge is brought about by the surge center, while the basal level of the GnRH is brought about by the tonic center. These are the potential regulators of GnRH pulses at puberty. Of course, we have the development of the surge center. This is very important, of course, in the female animal. And we also have the changes in the feedback sensitivity to estradiol. So it is said that immediately before puberty, the feedback sensitivity to estradiol decreases. This diagram shows the development of the hypothalamic GnRH surge center in the female animal following ovarectomy. So this experiment is conducted again in ovarectomized female sheep or ewe. And this number here represents the age of the animal: 3 weeks, 7 weeks, 12 weeks, 20 weeks, 27 weeks, and adult. The puberty in the ewe occurs at about 28 to 30 weeks. If we look over time by administering the estradiol implant, even though they do not have ovaries because they are ovarectomized, they eventually develop the ability to release GnRH and LH way before puberty. So again, the puberty in these animals occurs at about 28 to 30 weeks. But in here, after the implantation of the estradiol, the animal has gained the ability to produce GnRH even before the puberty occurs now at 27 weeks, 27, 28 to 30 weeks. So this suggests that the surge center is already functional at this point in time, now before puberty has begun. From this experiment, we can infer that the estradiol has a positive feedback effect. So implantation of the estradiol um, in overrectomized ewe before puberty induces or causes the surge of GnRH as shown in the diagram, a surge in the GnRH and a surge in the LH. So of course, we define a positive feedback loop in the endocrine system. Uh, this occurs when the release of a hormone initiates the actions that lead to an additional release of that hormone. In this case, we have the estradiol sending a positive feedback for the production of another GnRH and LH. Another factor that determines the onset of puberty is the changes in the feedback sensitivity to estradiol. So this diagram shows the changes in the feedback sensitivity to estradiol for ewe or sheep that have been ovarectomized. So ovarectomy at this particular age in weeks resulted in a drop in the level of the LH. Implantation of the estradiol at this time and the following weeks have resulted in a negative feedback for the production of the LH as shown in the diagram. So there is a decreased production of the LH. So at this part here, the level of the LH is constant. The drop in the LH is again brought about by the negative feedback of estradiol. So at 28 to 30 weeks as shown here, the LH level is beginning to increase to a constant level of E2. So the level of estradiol that is given to the animal is constant, but what at the point where the animal reaches 28 to 30 weeks, the level of the LH gradually increases. And this is because of a decreased negative feedback sensitivity to the effect of estradiol. We also have here the top portion. In the top portion, we have here the red part represents the proportion of the animals that have reached puberty or begin to cycle at this particular age. So at the age of about 28 to 30 weeks, there is 28, 30, 32 weeks, there is a gradual increase in the number or the proportion of animals that have cycled. So that at the age of about 34 weeks, the proportion of the animal that have cycled have reached about 75%, and at about 40 weeks no, or 39 weeks, so there is a 100% a proportion of the animal that have cycled. This also represents the uh, LH level that are continuously increasing as the animal increases its age. This diagram shows the changes in the feedback sensitivity to estradiol at different stages of puberty in the female animal. So in this case, we have here three stages: the prepubertal, pubertal, and the postpubertal, or that is after puberty. So this diagram here, this portion here represents the sensitivity to estradiol and inhibition of tonic LH secretion. So as you can notice here, before puberty, there is a high level of sensitivity to estradiol inhibition of the tonic LH secretion. So this means that before puberty, the influence of estradiol to LH is higher now because it has a more power to inhibit the production of LH. When the animal approaches puberty or when it is during puberty, there is a decreasing feedback inhibition of estradiol to LH secretion as shown in the diagram. And of course, right after puberty, there is a low level of feedback inhibition to LH secretion. That is why during the LH secretion, we can see the LH surge. So again, going back now to this to the portion at the bottom, for the prepubertal phase, the LH is minimal. There is also a level, the E2 or the estradiol is also produced minimally. And again, the E2 has a negative feedback to LH secretion. That is why there is a drop of the LH at this point. At puberty, as you can see here, there is a decreasing negative feedback of E2 on LH so that there is a pre-ovulatory LH surge. There is also an increase in estradiol at this point. This is very important for ovulation to occur. After ovulation, the CL will form, and the CL is important for the P2 production or the progesterone production. And of course, the progesterone, as you can notice here, it has a negative feedback. It always has a negative feedback to the release of GnRH, so that as long as the corpora lutea or the CL is present and the progesterone is produced, we do not get GnRH surge. When the CL regresses, as you can see here, there is also a decrease in the progesterone. There will be a decrease in the negative feedback on the GnRH surge. That is why you can see here another surge in the GnRH and LH. This slide shows the silent ovulation at puberty. So this is the prepubertal phase. We have here the broken line represents the onset of puberty, and this is the pubertal phase. So there are, as you can see in the diagram, there are three LH surges, and of course the very first LH surge is not associated with estrus behavior. So an example of an estrus behavior is standing, that the female now standing to be mounted or standing heat. After the first LH surge, there is a small bump in the P4 or the uh, progesterone, meaning that there is the formation of the CL at this time, but it is not fully formed at this particular time. After that, there is another LH surge as shown in the diagram, but this is considered to be silent because again there is no external manifestation of behavioral estrus. At this point, we also have the formation of the CL so that the P4 or the progesterone also rises. And we also have the next. No? After that, there will be another LH surge. If that another LH surge, and this is the one that is associated with estrus behavior. So this is actually the first estrus of the animal. And of course, there is already the manifestation of estrus at this time. So again, the silent ovulation is the first ovulation which is not accompanied by behavioral estrus. This is particularly seen in heifers in ewes. So thus the age at first estrus may not reflect the true acquisition of puberty. This diagram shows the influence of the steroid hormone on the GnRH neurons. As in the diagram, the GnRH neurons are the one that produces the GnRH in the hypothalamus. So the GnRH will synapse with the Kisspeptin neurons, and within the Kisspeptin neurons, there are certain receptors where estrogen and progesterone will be able to bind. We also have the Kisspeptin neurons that have a receptor, and at this receptor, these receptors can have positive or either negative feedback to the GnRH secretion. So as shown in the diagram, we have the Kisspeptin neuron, the one in green. We have the receptor for estrogen, and the estrogen can have a positive or negative effect or feedback. The negative feedback will uh, have also a negative effect on the GnRH neuron because it will only release a small amount of GnRH. For the positive feedback from the estrogen, it will send a positive feedback to the GnRH neurons to produce more GnRH. So there are certain stimuli from the environment or from um, the metabolic signals that can send a positive or negative feedback to the Kisspeptin neurons and to the GnRH neurons.